A company fed an AI 2 billion molecules, and it found a working antibiotic overnight, something no human had ever thought to try. In 12 hours.
Not 12 years, not 12 months, 12 hours. No sleep, no grant committee, no 3-year peer review cycle, no departmental politics.
Who owns the patent? If an AI generated the molecule, I need to know who I'm buying equity in before the market opens.
Dr. Silly, is the AI going to replace all the chemists? Are we all just living in a pharma simulation now?
My uncle tried a new medicine once.
But here's what nobody is asking. If it found one antibiotic overnight, what's stopping us from training it on every molecule that has ever existed and just generating all of them, the entire chemical universe?
The laws of chemistry, largely.
Mildly inconvenient.
That is my line.
Stop. We are not talking about overnight miracles, or AI replacing chemists, or whatever patent claim Crabzilla is building on a napkin right now. Before we can talk about any of that, we need to understand one thing. Drug discovery is not about finding a magic molecule. It is a pipeline. Target identification, hit generation, lead optimization, ADMET profiling, clinical trials. AI is not replacing that pipeline. It is being inserted into every single stage of it. By the end of this episode, you will understand exactly where AI fits, how models like variational autoencoders, graph neural networks, and large language models are actually being used, and why the real revolution is not speed alone. It is the ability to search a chemical space so vast that no human team could ever navigate it unaided. Welcome to the Krusty Lab. I am Dr. Seeley.
I'm Bobby Sponge, and I have never been more nervous and excited simultaneously in my life.
I am Kelpie. I have a variational autoencoder running on my GPU cluster in my shed, and it has been training for 6 months, and the results are, I'll talk about it later.
Crabzilla. I sent 4 emails before we started recording. 2 of them were to venture capitalists.
Squiven. I have reviewed the literature. It is mildly extensive.
Patar.
Today we are going deep into artificial intelligence in drug discovery. Not the hype version. The actual mechanisms, models, and methods. Let's go.
If you don't subscribe right now, I am going to train a model specifically on the behavioral signatures of people who didn't subscribe and use it to generate targeted molecular interventions. I'm calling it RegretNet. The architecture is almost done.
To understand what AI is doing here, you need to understand what drug discovery actually is. It is not find a molecule, take it, feel better. It is a multi-decade, multi-billion-dollar pipeline with a failure rate above 90%. The average drug takes 12 to 15 years to reach patients and costs somewhere between $1 and $2 billion. Most candidates fail not because the science was wrong, but because we could not predict early enough which ones would fail.
12 to 15 years? That is longer than I have been alive by a significant margin.
Which is precisely why I am invested in this space. The inefficiency is the opportunity.
It is mildly concerning when someone describes a decade of patient suffering as an under-optimized market.
I prefer friction-heavy TAM.
The pipeline breaks down into roughly 5 stages. Stage 1, target identification. You find a biological target, usually a protein, that, when you modulate it, treats the disease. Stage 2, hit generation. You screen millions of molecules to find ones that interact with your target. Stage 3, lead optimization. You take your best hits and chemically modify them to improve potency, selectivity, and safety. Stage 4, ADMET profiling. Absorption, distribution, metabolism, excretion, toxicity. And Stage 5, clinical trials.
And AI is showing up at all of them.
Every single one. The question is how and why it matters at each stage.
Wait, but where does the AI actually live in this? Is it like a robot in a lab coat somewhere?
Sometimes things are just software.
Correct. AI in drug discovery is computational. Neural networks, language models, generative models, operating on molecular data, protein structure data, genomic data, and clinical records. The chemistry still has to happen in the real world, but AI is compressing the search dramatically.
Okay, I need to say this first, because people genuinely do not appreciate how insane the scale is. The number of drug-like molecules that could theoretically exist, small molecules, the kind you'd put in a pill, is estimated at 10 to the power of 60.
How many is that actually?
More than the number of atoms in the observable universe, by many orders of magnitude. The entire observable universe contains roughly 10 to the 80 atoms. Chemical space is larger than that.
Excuse me.
So the total addressable market is effectively infinite. That's a good number.
It is mildly impressive that you framed a fundamentally unsolvable enumeration problem as a business development opportunity.
The chemical space problem is exactly why traditional high-throughput screening, where you physically synthesize and test hundreds of thousands of compounds in robotics-driven assays, is inherently limited. Even a cutting-edge screening campaign tests maybe 10 million compounds. That is not even a rounding error in a space of 10 to the 60. AI can navigate a much larger space by learning the underlying structure-activity relationships, the rules that map molecular structure to biological effect, and using those rules to search intelligently rather than exhaustively.
So instead of trying every key, the AI learned what a door looks like?
That is a reasonable analogy, yes.
A very large soup with too many ingredients.
That is less precise, but we can work with it. Now let's get into the models. The first class I want to explain is the Variational Autoencoder, the VAE, because it is particularly elegant for this problem.
Tell them about the latent space, please.
The core challenge is representation. A molecule is not naturally a number. It is a graph, atoms as nodes, chemical bonds as edges, or it is a smile string, a text-based notation like C1, CCCCC1 for cyclohexane. To use machine learning on molecules, you need to encode them into a format a model can work with, a high-dimensional vector in what we call a latent space.
Latent space sounds like a place where all your feelings go when you pretend they don't exist.
Suppressed emotions aside, a latent space is a compressed mathematical representation of the molecule. A VAE learns two things: an encoder that maps molecules into this space, and a decoder that maps points in that space back into valid molecules.
And here is where it gets magical. The latent space is continuous and smooth. Similar molecules cluster together in nearby regions, which means you can do arithmetic on molecules. Move in a direction in latent space, and you are literally interpolating between chemical structures. You can navigate the space rather than enumerate it.
You can do math on drugs?
We should patent the coordinate system. Molecular GPS. Series B, minimum.
The coordinates belong to the embedding. The embedding belongs to whoever trained the model. I would strongly recommend consulting a lawyer before Friday.
I have 3 on retainer. Different jurisdictions.
The VAE is trained on large databases of known molecules, Chembl, Zinc, PubChem, containing tens of millions of compounds with known properties. The encoder learns, given this molecule, what region of latent space captures its essential chemistry. The decoder learns, given a point in latent space, reconstruct a valid molecule. Then during generation, you sample new points, especially in regions associated with desired properties, and decode them into entirely new molecules that have never been synthesized.
So the AI is designing drugs that don't exist yet?
Generating candidate structures, yes. Whether they actually work is a separate and still expensive question.
A question you answer by synthesizing them, which I have absolutely been doing in my shed.
That is mildly illegal depending on which compounds are involved.
3 jurisdictions have weighed in on that question in different directions.
Once you have generated candidate molecules, you need to predict their properties before you commit the resources to synthesize them. This is where graph neural networks, GNN, come in. A GNN operates directly on the molecular graph. It performs what's called message passing. Each atom gathers a message from its bonded neighbors, aggregates those messages to update its own representation, and passes the updated representation back. After several rounds of message passing, each atom's embedding captures information from atoms several bonds away, the local and global chemical environment simultaneously.
It's like each atom texts its neighbors about what's going on and they all build a group chat?
That is approximately how message passing works. Yes.
The wild thing is these models generalize. You train on known compounds and they make reasonable predictions about molecules they have never seen, as long as those molecules are not too far from the training distribution. Archie, push too far out into unexplored chemical space and accuracy degrades fast.
What if the weird molecules are the ones that actually work?
That is genuinely one of the core unsolved problems in the field. The exploration-exploitation trade-off in generative drug design. You want to explore novel space, but your models are calibrated on known space.
It is mildly ironic that the models are most uncertain precisely where the most novel discoveries would be found.
You don't know what you're looking for until you find it.
Patar, that is actually a precise description of the de novo drug design problem.
I was thinking about mushrooms in a field.
The analogy survives the context.
Okay, we cannot skip the LLMs. People think large language models are just for generating emails and summarizing documents. They are being applied to drug discovery in ways that are completely unexpected, and some of them are working.
Not unexpected. Logical. Large language models trained on scientific literature and on protein sequence databases can perform a kind of knowledge synthesis over mechanisms, drug interactions, genomic associations, and clinical observations that would take a team of researchers years to assemble manually. Models like Galactica and BioMed-LM have demonstrated meaningful performance on biomedical reasoning tasks, predicting gene disease associations, summarizing mechanism of action from literature, even proposing novel target hypotheses.
Wait, you can just ask an AI about which proteins to target?
With appropriate skepticism and validation, yes. But there's a more interesting application. Models like ESM2 from Meta are protein language models. They treat the amino acid sequence like a text. Each amino acid is a token, and learn the statistical structure of protein sequences across hundreds of millions of known proteins. From these patterns alone, without explicit biochemical instruction, they learn representations that capture secondary structure, evolutionary conservation, and functional sites.
Which means you can use the model not just to understand existing proteins, but to generate new ones with desired properties. Prompt the model in the right direction, and it can sample amino acid sequences that fold into functional proteins. This is how AI-designed enzymes and antibody fragments are being produced.
Wait, you can generate a new protein?
Functional novel proteins have been experimentally validated. Protein MPNN, RF diffusion, ES fold. Real tools, real outputs, real synthesized and tested proteins.
Is that a service? Protein GPT, freemium tier for soluble proteins, pro tier for membrane proteins, enterprise tier for anything that might be immunogenic.
It is mildly inevitable that this service will exist within 18 months.
I'm filing the trademark today.
I've been fine-tuning a protein language model on my GPU cluster. My electricity bill it's become a conversation between me and my electricity provider. But I'm not stopping.
It is mildly surprising that Kelpie's shed has a GPU cluster.
It has 3. The neighbors believe I am mining cryptocurrency.
That is mildly preferable to the accurate explanation.
We need to talk about AlphaFold because you cannot discuss AI and drug discovery without it. For decades, determining the 3-dimensional atomic structure of a protein required X-ray crystallography, cryo-electron microscopy, or NMR spectroscopy, all expensive, slow, sometimes impossible for certain protein classes. Many medically important proteins resisted structural determination for years. AlphaFold 2, released by DeepMind in 2021, predicted the structure of essentially every known protein with near experimental accuracy, from nothing but the amino acid sequence.
You were telling me we spent decades using massive, expensive machines for individual proteins, and an AI just did all of them?
The AlphaFold protein structure database now contains over 200 million predicted structures, covering essentially the entire known proteome across all sequenced organisms.
200 million. Done? The whole thing.
So the bottleneck just shifted downstream. Interesting. I'm repositioning my portfolio as we speak.
The relevance for drug discovery is this. Structure-based drug design depends on knowing the 3-dimensional shape of the target protein so you can design a molecule that fits into its binding pocket. Previously, this approach was limited to proteins with experimentally determined structures. Now you can do structure-based design against almost any protein with a known sequence. The search space for potential drug targets has expanded by orders of magnitude.
So AlphaFold made a map of every possible lock, and now we can design keys for all of them?
Yes, that is what happened.
I understood the whole thing. This is genuinely one of the best days of my life.
A map is useless if you don't know where you want to go.
She is right about that, too. Knowing the structure doesn't tell you whether inhibiting that protein will treat disease, or whether the binding pocket is druggable at all, or whether a molecule sitting in that pocket will have catastrophic off-target effects. The structure is necessary, but very far from sufficient.
Okay, diffusion models, I have to talk about this because this is where the field is getting genuinely feral.
Go ahead.
Diffusion models, the same class behind image generation systems like Stable Diffusion, are now being used to generate molecules directly in three-dimensional space, not as text strings, not as graph encodings, as actual atoms placed in three-dimensional Cartesian coordinates. Models like DiffSpeed generate molecules directly inside a protein-binding pocket, conditioned on the pocket geometry. The model learns to denoise random atomic positions into valid synthesizable molecules that are geometrically complementary to the target site.
The AI is drawing molecules in 3D space inside a protein?
In 3 dimensions, with physically correct bond geometries, shaped specifically to the binding site. It is generative art, but for chemistry and the outputs are being experimentally tested.
This is the same creative AI content market, but for atoms. I wonder if there's a collab opportunity with a design platform. Your pocket, your molecule, your brand.
That is mildly the most reductive framing of structure-based drug design I've encountered.
I'm going for accessible.
The technical challenge with diffusion models in molecular generation is enforcing chemical validity. An image can have artifacts. A molecule with invalid bond angles or impossible valence states cannot be synthesized. So these models are being extended with SE3 equivariant architectures, meaning they respect rotational and translational symmetry in three-dimensional space, and chemical validity constraints baked into the training objective.
What does equivariant actually mean here?
It means the model knows that rotating a molecule in space does not change what the molecule is. A carbon atom bonded to 3 hydrogens is the same atom regardless of which direction you're looking at it from. Models without this inductive bias waste enormous capacity learning that basic symmetry from scratch. Equivariant models get it for free by construction.
So we built the laws of physics into the model itself?
The relevant symmetries of physical space, yes. That is exactly what equivariance means in this context. Let's return to ADMET because this is where most drugs actually die. You can design a molecule that binds beautifully to its target and still fail in the clinic because it is insoluble in plasma, because it is metabolized by cytochrome P450 enzymes before it reaches its target tissue, because it inhibits the Hurgy potassium channel and causes fatal arrhythmia, or because it is hepatotoxic at therapeutic doses.
The graveyard of drug discovery is full of compounds that killed cancer cells perfectly in a dish and then destroyed the patient.
Precisely. AI is being trained on large ADMET datasets. Hurgy toxicity panels, CACO2 permeability assays, metabolic stability measurements, plasma protein binding fractions, CYP inhibition profiles, to predict these failure modes computationally and early. If you can filter out ADMET failures during the generative stage, rather than after synthesis, animal testing, and early phase trials, you save years and hundreds of millions of dollars per program.
So the AI can screen out the bad ideas before anyone even makes them?
That is the goal. And increasingly, ADMET models are being integrated directly into the generative loop. Instead of generating molecules and then checking properties as a filter, you generate molecules under multi-objective constraints, optimizing simultaneously for predicted target binding affinity, metabolic stability, solubility, and safety flags. The generation and the evaluation happen together.
So you're collapsing 12 years into what? 18 months?
The timeline compression is real but more modest than headlines suggest. The computational discovery phase has accelerated dramatically, but clinical trials are irreducibly slow. Biology is the bottleneck, not computation. A phase 3 trial takes the time it takes because human outcomes take the time they take.
It is mildly refreshing when someone on this program acknowledges that the bottleneck is not always the part AI touches.
Okay, but the clinical trial design is where we should insert AI next. So, adaptive trial designs, Bayesian response adaptive randomization, synthetic control arms built from real-world evidence.
That is a topic for a different episode.
I'm available every week indefinitely.
Let's ground this in specific cases. In silico medicine, used a generative AI pipeline, target identification by deep learning on multi-omics data, candidates generated by a generative model, lead optimization by reinforcement learning, to discover a novel TNIK inhibitor for fibrosis in 18 months from target identification to preclinical candidate. For context, that stage typically takes 3 to 5 years.
18 months instead of 5 years.
Exscientia designed a compound for obsessive-compulsive disorder using AI-assisted molecular design that entered phase 1 trials in 12 months, roughly 4 times faster than industry average for that stage. And the Halison story Kelpie mentioned at the start. A neural network at MIT trained on existing antibiotics identified structural signatures associated with membrane disruption, applied that model to screen over 100 million compounds computationally, and flagged Halison, a structurally unusual molecule that kills drug-resistant bacteria, including Mycobacterium tuberculosis and pan-resistant Acinetobacter baumannii. It had been synthesized before, but never evaluated as an antibiotic.
Named after HAL 9000?
It is a deliberate nod to the AI connection by the researchers.
That is so genuinely cool and also slightly ominous, and I cannot decide which feeling is winning.
I have been attempting to replicate the Halison discovery pipeline in my shed. I have the zinc database, I have a GNN trained on the E. coli growth inhibition data. I do not currently have access to the relevant bacteria, for reasons that are largely legal, but partly practical.
Several reasons, I would imagine, in multiple layers.
The antibiotic market is worth $50 billion globally. If Kelpie's shed produces even a lead compound, that is a conversation about valuation.
It is mildly concerning that Kelpie's biosecurity situation is being evaluated as a fundable asset.
The honest picture: AI in drug discovery is transformative, but it is not magic, and the limitations are real. Distribution shift is a major problem. Models trained on known chemical space can fail badly on truly novel compounds that sit far outside the training distribution, and those are often exactly the compounds most worth finding. Synthesizability is a bottleneck. A generative model can produce a molecule that looks excellent on paper but has a synthetic route requiring 17 steps, exotic reagents, and reaction conditions that do not exist at commercial scale.
I have generated beautiful molecules that, when I actually sat down to plan the synthesis, required a reagent that is manufactured by one company in one country with a 6-month lead time. That is not a drug candidate. That is a thought experiment.
Which is why synthesizability-aware models like ASCOS are being integrated into the pipeline. ASCOS does retrosynthetic planning, starting from the target molecule and working backwards through known reactions to commercially available starting materials, and can evaluate whether a generated molecule is actually makeable. You evaluate candidates not just on predicted potency, but on predicted synthetic accessibility simultaneously.
Retrosynthetic planning sounds like being given a cake and working backwards to figure out what groceries you need.
That is accurate, yes. You know what you want to make. You work backwards through the reaction network to find a feasible starting point.
And then you work out whether you can actually afford the starting materials, which is the part that killed 3 of my projects this year.
Sometimes you plan a very complicated dinner and then remember you can't cook.
The AI can cook. It just sometimes invents recipes that require ingredients that don't exist yet.
There is also the regulatory question. The FDA has issued guidance on AI in drug development, but the question of how you validate an AI system's predictions, how you establish that a model is reliable enough that its output should influence the design of a human clinical trial, is not solved. Explainability, reproducibility, model governance, data provenance. These are genuinely hard problems that the field is actively working on.
So we have AI that can find drugs, but we don't always know if we can trust it?
We can trust it when its predictions have been experimentally validated across a relevant chemical space. The challenge is knowing when to trust it in novel regions where no experimental data exists to calibrate against.
That gap between model output and validated confidence is itself a market. I am seeing a pharmaceutical AI auditing service, Drug Audit Pro, licensed per molecular program. I will take a meeting with anyone in the space.
It is mildly inevitable that this service will exist. It is also mildly concerning who will be running it.
I'm available.
That is what I meant.
Okay. PATAR. We have covered variational autoencoders, latent space, graph neural networks, protein language models, AlphaFold, diffusion models, ADMET prediction, retrosynthesis, and the regulatory landscape. What do you make of all of that?
You have too many molecules to look at. You cannot look at all of them. So you teach a computer to feel what a good molecule feels like, from its shape, from its neighbors, from the patterns of all the ones that worked before. Then you ask it to imagine new ones in the right direction. You still have to check whether they are real. But now, you know where to look.
PATAR. You just summarized the entire field of generative AI in drug discovery in 4 sentences. How? How do you do this every single time? I have been sitting here for an hour, and that is the clearest thing anyone has said.
That was genuinely the clearest summary of the core problem and the AI solution that I have heard in a long time. And I have a PhD in computational chemistry.
She's been listening the whole time and just synthesizing it perfectly in the background. PATAR, I want you on my research team. Full access to the shed.
That is mildly the most dangerous job offer I have witnessed on this program.
I would need my own parking space.
Final exam. This is happening right now. If you have been paying attention, this is your moment.
We built an entire episode around this. Do not let us down.
Pause the video, answer these 3 questions, then press play. Question 1. A variational autoencoder for drug discovery learns to encode molecules into a continuous latent space and decode points back into molecules. What specific property of that latent space makes it useful for generating new drug candidates? And why does that property matter more than simply memorizing existing molecules? Question 2. Graph neural networks model molecules as graphs with atoms as nodes and bonds as edges, and use message passing to build atom representations. What exactly is being passed in each message passing step? How does this capture chemical context? And how does the final molecular level prediction work? Question 3. AlphaFold 2 transformed structure-based drug design. What does it predict? What information does it use as input? And what specific aspect of its training, beyond just large-scale deep learning, explains why it achieves near-experimental accuracy? We will wait.
Take your time.
I will be running a hyperparameter sweep.
Okay. Question 1. The latent space is continuous, so nearby points correspond to similar molecules, and you can sample new points, especially points in regions associated with desirable properties, and decode them into molecules that have never been made. It's not memorizing. It's learning the underlying structure of chemical space and using that to navigate it.
Correct. The continuity means interpolation between known molecules produces chemically plausible intermediate structures. And critically, you can condition the sampling, bias it toward regions of latent space where a property predictor says the properties are good, so you are not searching randomly. You are searching intelligently toward desired outcomes.
Bonus, you can also optimize directly in latent space. Encode a known molecule. Follow the gradient of a binding affinity predictor through the latent space and find structurally similar molecules with better predicted activity. Bayesian optimization over the latent space is a major research direction.
Accurate and worth knowing.
The latent space is navigable real estate. Location, location, location.
It is mildly exhausting how reliably you reach for property metaphors.
Question 2. Message passing sends each atom's current feature vector to its bonded neighbors. Each atom aggregates the messages it receives from its neighborhood, updates its own representation, and after K rounds, each atom's embedding captures information from all atoms within K bonds. Then you read out a molecular level prediction by pooling all atom representations, summing or averaging, and passing that through a final prediction layer.
Precisely right. The key insight is that the receptive field expands with each layer. A 1-layer GNN captures local bonding environment. A 3-layer GNN captures structural context, 3 bonds out in every direction. Deep enough networks can, in principle, capture global molecular topology, ring systems, functional group interactions across the molecule.
I got it. I actually got it.
I missed question 3 because I was adjusting learning rates. But AlphaFold 2 predicts the 3-dimensional atomic structure of a protein from its amino acid sequence. It uses an architecture called evoformer that processes 2 inputs simultaneously: the amino acid sequence of the target protein and a multiple sequence alignment, a comparison of that sequence against hundreds of evolutionarily related sequences across different organisms. The evolutionary information is critical. Positions in the sequence that are conserved across millions of years of evolution tend to be structurally or functionally important. Positions that co-evolve, meaning when one mutates, another tends to mutate too, are often in physical contact in the folded structure. AlphaFold learned to read those co-evolutionary signals as geometric constraints on the fold.
Comprehensive. The coevolution point is exactly right and is the reason AlphaFold works as well as it does. The model is not predicting structure from physics alone. It is reading the evolutionary record of what worked.
I said AlphaFold looks at the protein very carefully. I think that was technically true, but missing some detail.
Considerably some detail.
I answered all three in terms of market disruption potential. I believe at minimum one should count.
None of them count.
It is mildly predictable that the quiz results mapped exactly onto each person's fundamental epistemology. What today's episode demonstrated is not that AI is replacing drug discovery. It is that AI is changing the shape of the search problem. The pipeline is the same, the chemistry is the same, the biology is the same. What changed is the ability to navigate a space that was previously too large to search in any systematic way. That is not a small change, but it is a precise one.